Professor Daniel Buchan. He's a, pardon for the mispronunciation if it's the case. He's an assistant professor at UCL in London, where he develops machine learning tools for the analysis of protein structures and function. His expertise spans research, software engineering, machine learning, and the development of online platforms for high-throughput biological data analysis. Interestingly, before his current role at UCL, he worked as a data scientist and analytics manager in industry. And today he's going to present one of the tools that he developed for protein analysis. So Daniel, please, the floor is yours.
Sure, great. Yes, thank you for the introduction and thank you for the invite. So yeah, as mentioned, I'm Daniel Buchan. I'm the lecturer in bioinformatics in the UCL Computer Science Department. I'm going to talk to everyone today about what we call the PSIPRED Workbench. So this is actually a number of tools that we make available to researchers online. So just to give you a little kind of heads up, structured this roughly into 3 sections. So I'm going to talk about what is PSIPRED. So it's 2 different things, as we'll find out in a second. Then I'm going to talk about new methods, new predictive tools that are available on our web services. So not some of the older ones that I think we're more famous for, but we'll cover one of those. But mostly the kind of the new things and the things that we think are most exciting at the moment. And then I'm just going to finish giving you a little overview of some current work that includes some of the synthetic biology stuff that we are starting to get into these days. Cool.
Sorry, before we start, we see a little window with all the pictures of the participants in the video call that cover part of the slides.
Yeah, I can hide that.
That's perfect.
Yeah, sure. Yeah, so is there a way to hide that? Just stick it in the bottom. Okay, cool.
Yeah,
okay,
that is perfect.
Okay, so PSIPRED is kind of 2 things. So PSIPRED, the algorithm. So this is a method that was developed by Professor David Jones, also here at UCL, around 1998. And depending on what point in time you look at it, it's either the most or the second most accurate secondary structure prediction algorithm out there. And so what we do is we take an amino acid sequence, we do a search of a sequence database to build an MSA, and then we use a neural network to try and predict the secondary structure for that sequence without doing any 3D modeling. And what we essentially output is for each residue, the assignment for whether it's in the helix. So in my diagram here on the left, these are the nice kind of pinky cerise helices. So whether a residue is part of a helix, whether it's part of a beta strand, so the yellow kind of arrows, or whether it's just neither of those. So we just assign those to coil. So this is a 3-state secondary structure prediction. The first iteration of the algorithm in 1998 had an accuracy of 85%, which I think was the leading accuracy at the time. The current iteration of the algorithm, version 4's accuracy, I think, is 94.7%. It's kind of approaching the total amount of information content that you can actually extract from an MSA. So I don't know that you can really do much better than that. So that's the algorithm. And then, so Professor Jones, back in 98, I think he was at Warwick University, decided to make a little web server so that if you were a biologist, you could come and do a secondary structure prediction. You wouldn't have to install this software and things. So he started off the first PSIPRED web server. And since then, it has kind of grown to add more and more predictive methods. So we now call it the PSIPRED Workbench. And so we offer a suite of 18 different, mostly machine learning-based predictors, analysis tools for analyzing structure, protein sequence. And we cover quite a very wide variety of different predictors. So we can predict the location of membrane helices. We can take a structure and decompose it into structural domains. We can take a sequence and find the main boundaries. We do function prediction by predicting Go terms. We can predict metal binding residues, both on a sequence basis and a structural basis. There's disorder prediction, membrane helix packing prediction. Quite a wide variety of different ways that you can kind of characterize this functional features or the structural features of proteins. So that's kind of what it's grown into. In 2018, I kind of led on completing a very large project to kind of completely retool all of our web servers, rewrite it, make it much faster, make it more user-friendly, and so forth. And so these are our usage stats roughly since then. And so I think you can kind of see the size of the bar is the number of analyses. And we do roughly somewhere between 250,000 to 300,000 kind of predictions for researchers around the world. And you can kind of see from 2019, 2020, 2021, the number of analyses growing, because I think because it was faster, it was more user-friendly. Then there's a sudden marked step down in 2022, and this is the year that AlphaFold 2 was released. And I think lots of people kind of, felt, why do we need to do any of these predictions? Lots of people might have just said, we'll just predict the structure and that's enough. Since then, we've seen our kind of server usage kind of rebound quite a bit. And in the end of the day, an AlphaFold structure is itself a prediction. It's not always right, no matter, even though it's very, very good. And so we've seen, I think, lots of people want to still do corroborating predictions. They want to do consensus predictions for things. The colors on the chart here, these are the different methods. And again, I don't have time to go into all of them, but suffice to say the kind of pale green, the secondary structure prediction is our most popular method. It accounts for nearly sometimes more than 2/3 of the predictions we do each year. If you're curious who's using the service, we have this map of the world. It's just colored, so darker is more usage in that country and lighter is less usage. And we kind of just see the expected kind of pattern of usage with the USA, China, India, Japan, the UK, Germany being the most common, the countries that make most use of our service. So, okay, I've got a very brave, perhaps live demo here. So this is the website itself. So just saw a picture. And essentially at the top here, you can pick whether your input data is a protein sequence, a protein structure. We have kind of 5 methods here. We've got a new method for RNA-seq analysis that we'll talk about in a bit. But just the sequence data is, I think, quick and easy. So you would pick the methods. So you can pick one or more methods. I'm just going to select the secondary structure prediction. I'll just zoom in a bit so that's a bit easier to see. Then at the bottom, there's an input for your sequence. So we've got a handy test sequence you can auto-load. We ask that you give your submission a name so that you can keep track of it and we can keep track of it. Optionally, you can give it an email. I mean, I suggest you do give it an email because then we'll send you a little email reminder. And then you press submit. And you get presented with the eventual results page. So we just echo back your sequence to you, and eventually we'll fill this in. And we could wait for 3 to 5 minutes. So luckily, I've prepared one earlier. And so this is just the result we'd have got if we'd have waited. And here we're just coloring up the residues depending on the assignments that we made for our different predictors. And so in gray, just coil, unassigned residues. In pink, this kind of cerise color like the previous diagram. That's the alpha helix, and then in yellow, some beta strand. And usually we've got some ancillary other diagrams to present, or sometimes some charts and bits and pieces. And so we have this kind of cartoon that also presents the same data, but also with a kind of confidence score laid on top of that. And up here on the right, there's kind of various downloads. So everything here is essentially built out of the output files, the results. And so we have a little downloads panel where you can download the results in text format, import that into the program or Excel of your choice. Cool. So how do we get access to the server? As I've just shown you, we've got our website, so fairly straightforward. In the last handful of years, we have a new YouTube channel. So all of our most popular methods and our most recent methods have a little tutorial video, and that will walk through what data you have to input. And then we'll walk you through interpreting the results, like what are the different diagrams, tables of numbers, confidence scores, what do they mean and how should you interpret them? And also they'll walk you through, if you download the plain text results, just walk you through what the format for those are. They're mostly CSV files with some description, but yeah, it's worth knowing. If you're in need of any help, we have an email address. So if there's a problem with the server or you just need a bit more help in interpreting it, we're usually quite happy to answer these emails, sometimes not as fast as we like. Sometimes fail to notice a help email, but we're trying to get around to all of them. And lastly, all of the code is available. So every single method we make publicly available as code that you can download and run yourself. Even the web server, if you wanted to run a copy of the web server yourself, you can download all that code. So you can go to our GitHub page and you can go through our various methods. There are some methods that we make accessible that aren't on the web server because they're just too complicated or they're too long running that it makes more sense for users to download the code and run it themselves. Anything that is available has an Ansible installer, so it should be fairly straightforward. You can download the package, you can download Ansible, then you can just run for each method this Ansible playbook install command, and it should install the software. So that's the web server if you haven't previously seen it. So now I'm just going to talk about some of the new methods, the predictive methods that are available that we've kind of updated in the last handful of years. So there's kind of these 6 different methods that I think are probably the most interesting of our kind of new developments. So DMP form 2, this is a protein structure predictor. And so kind of, I hope you can see the arrow, my kind of pointing arrow. And so starting in the top left here, you generate a multiple sequence alignment for your input sequence. And then you're going to kind of calculate some embedding vector that we're going to use for the prediction. And I think it's worth me spending a couple of slides talking about what embeddings are, just in case you're not used to this in machine learning, just because all of the methods pretty much I'm going to talk about, I'm going to say embedding a lot. And so to my mind, an embedding is a way of building a neural network that can learn some transformation of some high dimensional data, often categorical data, to some reduced dimensionality representation, but that reduced dimensionality representation preserves all of the information we need to make some prediction, right? And then once we've learnt this embedding method, we can then use that such that we can use our lower dimensional output to make our predictions. And so the classic example of this is kind of text analysis, so the kind of thing that ChatGPT does. So if we think of the sentence, the king was exceptionally wealthy, we have like 5 tokens, so 5 words, and we want to know the relationship, want to learn the relationship between the words such that we can learn how to kind of embed the information here into some low dimensional space. And of course, we're comparing words across sentences. So we can have the sentence, the king was exceptionally wealthy and the queen was exceptionally wealthy. And what we can see from that ourselves is that king and queen are 2 words that can function in the same context, right? The context exceptionally wealthy. So it can go with those words. But we wouldn't expect a word like beggar to go in that sentence. Like the beggar was exceptionally wealthy is going to be rare in our training data. Now, if we think about a naive way of representing sentences in machine learning, let's say all of our sentences are 5 words long. We've got a vocabulary of 9,000 words. So each sentence, might require a matrix that's 5 by 9,000 in size. It's a very big 2-dimensional matrix. And so what we want to do is learn a way of compressing that matrix down to some smaller representation. And so the classic one in language, people show you these 2D projections. And so now we've taken our 5 by 9,000 matrix and we've compressed it down to 2D. So each word has some 2D location in this space. And we can see in this particular space over here on the right, king and queen are next to each other because they're words that can be used in the same context. They get projected to a similar region in the space. And that's essentially what we're learning when we're learning some quote unquote embedding. So when we start thinking about proteins, like a simple way to think about proteins is that each amino acid is a word in a sentence. And so our typical inputs for doing protein bioinformatics, a standard input is the length of the protein times, say, 20 or 22, depending on how you're handling the amino acids. So that's quite a large 2D matrix, potentially. A multiple sequence alignment, that's the length of the protein times by the number of rows, the depth of the MSA times by 22 again. So these are quite large, high-dimensional objects. And we usually want to learn some kind of output that we can handle more simply. And the standard sets of outputs are maybe a fixed length embedding. We just say, no matter how long the protein is, we're just going to have, say, 64 by 20 or 64 by 22. So we're always going to compress our big matrix down to this smaller matrix, 64 by 20, let's say. Another thing, a single vector embedding, no matter how complicated our matrix is, be it a protein or MSA, we're just going to output some single vector of 32 values. So that's like a single vector embedding. Or we can do something that's a kind of protein length embedding where we just say, no matter how long it is, it's only ever 32. the other dimension is only over some fixed dimension of some size. And so that's essentially what our DMP-fold method does. So up here in the top left, we have kind of 2 passes of some neural network that outputs a little 2D, fixed size 2D representation that represents the MSA. We then input what we call our precision matrix and what's initially a random distance map into our predictor. And if we follow down the right-hand track, we get to some C alpha coordinates for our proteins. And then we just kind of iterate. We put the C alpha coordinates back in at the top. We calculate a more correct distance map. The initial distance map is kind of random. And then we just keep iterating until the coordinates pass some confidence threshold that we're happy with. And so this is typically 3 iterations. When we hit that confidence value, then we calculate the remaining sidechain coordinates, we calculate the confidences, we output a structure. So in 2018, when we developed this, when Professor Jones developed this, this would probably be the best performing protein structure predictor on the planet, except this was also the year that AlphaFold 2 was released. And so we got completely blown out of the water by them. The 2, I guess, good points about DMP-Fold 2 these days, it's about 100 times faster than AlphaFold 2 for what's actually quite a modest reduction in accuracy. So it's pretty good, much faster. And in my experience, it's way, way easier to install DMP-Fold 2 than AlphaFold or OpenFold. So if those 2 things are important to you, then maybe DMP-Fold is useful to you. So kind of moving on from this, one of our other new methods for characterizing sequences is our metal binding site predictor. So this is a sequence-based predictor. And so kind of taking this diagram, on the left, we have an input sequence. We use a sequence structure protein language model, so PROT-T5, and we put the sequence in and it outputs the embedding vector. So what are the values for the embedding? And then we just use that embedding vector as a set of features for a fully connected feed-forward neural network that just predicts which residue, one at a time, whether or not it's a metal binding residue and which metal it might bind. This was part of an entry to the UniProt Metal Binding Site Challenge from 2022. And as we understand it, this is the best performing method by quite a large margin. We were told they would publish a paper about this, but that's never materialized. So who knows? But we decided this was good enough that we just went ahead and put it on the website while we were waiting for the publication to come out. Probably, I think, maybe one of our most interesting recent methods is our structure segmentation. So the input here is a protein structure rather than a sequence. And so over here on the left, you provide us with a structure, and the first thing we calculate is the distance map, the distance between the C alpha carbons. We then extract the sequence, so that's one of the 3 inputs, and we calculate these things called backbone frames. So the way AlphaFold 2's structure resolver, or the structure modeling bit at the end of AlphaFold 2, you get these little kind of structural triplets. They're just the kind of the angles between C alpha carbons. So we decompose the structure into these little backbone frames. Then we put all of this into AlphaFold's IPA encoder, and we use that as a kind of embedding generator. So it generates a set of features that we then have a trained masked transformer decoder. So this is the actual predictor that then predicts which residue is in which domain, essentially. And so it outputs these 3 kind of outputs. So the first is the domain mask. So this is just working along the sequence and assigning whether a residue is in a domain or not. So domain 1, domain 1, domain 1, domain 3, domain 2, and so on and so forth. Then we also have a parallel prediction for whether or not a residue is actually in a compact structural domain or not. So some residues are in disordered regions, some residues might be in a domain, but AlphaFold has failed to predict it. So we try and predict domain, non-domain. And then we also output a confidence for each domain. And so on the right-hand side, we have a comparison with a couple of the other methods. And so I don't know how used to AlphaFold structures you might be, but if you go looking through the database, when it recognizes a structure, the structure is extremely good, but there's an awful lot of kind of spaghetti where AlphaFold doesn't really know what to do with it. It either hasn't seen the domain or the region is disordered and doesn't really kind of know. So you get these kind of weird, hairy spaghetti balls on occasion. So this is one of these examples. And if we give this to the Unidoc method, it kind of divides the whole structure up into 2 domains. So we have a kind of red domain and this kind of purple domain in the background. And any of the loops that are attached to those domains, it also assigns to the domains. So it's not a particularly good predictor for this particular structure here. The sort method does a much better job on the domain, so it gets the domain right, but it still thinks there's two domains. So there's the core domain, structural domain that's correct, and all the rest of the sequence almost gets assigned to just a second domain. So it's kind of like everything else is just some second domain. DPAM is kind of quite confusing. So what it does, it gets the actual structural domain correct, but then every single loop gets assigned to its own domain. So it generates this kind of 15 domain prediction. And out of these comparators, Morizo, our method, is the only one that kind of gets the domain right. So it assigns the domain correctly and assigns only the domain residues to the domain, and everything is assigned as non-domain. And kind of, some more kind of aggregate statistics, just comparing these different methods. Essentially, we're just working out the ratio of how often we are correct on the structural domain boundaries in comparison to the Cath database, which I don't quite have enough time to go into. But you can see our kind of average performance is a little better than all the rest, with a lot better precision than any of the other methods. So once we had this method in hand, this was kind of a little after the time that AlphaFold DB was released by the EBI, and they've got 214 million predicted structures, lots with very high confidence predictions, and they've kind of got all sorts of things in there. So, as we've seen, you've got these kind of really hairy ball spaghetti things where there are definitely some well-resolved, probably correct domains in the center. And then you've just got a whole load of sequence that AlphaFold just doesn't really know what to do with. You've got other sequences where most of the sequence is really well predicted. It's probably all right, but they're maybe not as, the overall prediction is maybe not as compact as a real protein might be. And then you've got things that are basically okay. So, they might have large disordered regions and things like many eukaryotic proteins have some kind of disorder. And, if you're working with structures, you might want to know what the domains are here. And if you're a data resource like Cath, you don't have the resources to manually segment 240 million structural models. So we worked with the Cath database to develop a new resource called TED, where we take 240 million AlphaFold models, and we segment them into 365 million domains. So here we're running Morizo, as I just described. We're running Cath's own automated domain segmentation tool, and we're also running FoldSeek. And so we get a kind of consensus domain region by combining all three predictors. And then we can compile this into our TED database. And this is available as several terabytes of information online, or you can kind of browse it via our new TED web portal at this address down here at the bottom. And that kind of led us to thinking, like, it's all well and good kind of browsing that, but if you're actually working with structures, you probably want to be able to search for related structures or structures that have similar functions and so forth. So we developed another method on top of this called Morizo search. So this diagram, confusingly, it's easier to kind of start at the bottom, so track B. And so we take the structure database here on the left. So in this case, it's AlphaFold DB, but any structure database. And so we have this little neural network called fold class, which is an embedding method for protein structures. So you put a structure in and you get some vector out on the other side. And so we basically just put every single AlphaFold DB structure in it, and we developed a pre-calculated set of 214 million embedding vectors. And we just squish that all down into a ginormous binary blob that lives on a hard disk, and it's about 2 terabytes in size. And then we can use that as a kind of search database. We can search against it. So it's much, much quicker than doing structure alignment. Yeah? And so the top track is how the method actually works for the user. So at the top, we have a structure that the user might put in. We segment that using Morizo into its individual domains. We run fold class to develop the embeddings for those domains. And then we search the big binary blob to just find the closest vectors. It's just a vector-vector lookup. How far apart are two vectors? It's quite simple maths, really. And we just do a very simple nearest neighbor lookup, like what are the nearest vectors to the inputs? We rank those. There's a small amount of kind of selection statistics, but we essentially retrieve the top 20 results. We quickly use TM-align to structurally align only those 20, and then we present you with the results so that you can see which are the structures in AlphaFold-DB and Cath as well that best match your query. So you can kind of do some evolutionary analysis, functional analysis, and so forth. A thing that's nice about it is that we will show you structures that have the same domain architecture, so the same domains in the same order, but also the same domains in different orders or with inserted additional domains. So we'll present you kind of all of the structures that have the same domains, same overlapping subset of domains. So we've also got a new RNA-seq analysis method that's quite recent that we call GS-RCL, which, a bit of a mouthful, is Gaussian scRNA-seq contrastive learning. And what we asked is, can we distinguish cell types in RNA-seq data? And that RNA-seq data is extremely similar, it's very overlapping. And so this is a good use case for what's called contrastive learning. What contrastive learning asks is, can we learn a way to separate out the two classes or the multiple classes that we want to predict, even when the data is highly kind of overlapping? And this relies on a kind of data augmentation strategy. So this diagram, which I partially understand from my much smarter PhD student, Ibrahim. So the top track is the pre-training. So on the left here, tagged S, this is our prediction data set. So each of these points is an expression profile for a cell, and we've got two classes of cell in green and red. And we want to be able to separate these out and predict which of the cells are the red cells, which of the cells are the green cells. And the way the contrastive learning works is we need to create two views. And the way we do that is we generate some random Gaussian noise and add it to the data set. And so in the middle here, S1, that's one of the views that we've created. Then we just do it again. We generate some new Gaussian noise, add it back to the data set, and then we have these two different views of the same data. Then we have a little neural network that we train to do a kind of pre-training task. And the neural network is divided into two bits. So G is the encoder that learns how to separate the data. And then H is a prediction head that is just there to minimize what's called the contrastive loss. And once we've got this completely trained, that the loss bottoms out, we then kind of know that our encoder has learnt a way to separate out these two cell types. So at prediction time, and that's the bottom track here, we take our data set, we pass it through the separation encoder, and then we get this data set with the separation values. And so hopefully you can see in this bottom middle section that lots of the green marked, the cell type A, have been pushed away from the red ones. So when it was just one completely almighty overlapping blob, most of these green spots have been pushed very far away. So now we've got values in some data space that we could apply some machine learning to. So in our case, we just train an SVM, but this could be a neural network, I don't know, GD boost, any kind of easy classifier. And then we get a kind of classification boundary, so that we can say that, you know, cell type A, maybe our hemopoietic stem cells, are inside this region of the classification space, and all the other cell types are outside of that region. So that's essentially how this method works. And then we can kind of compare our performance to some other methods. And so here, the charts on the left, our method is on the y-axis. And then the methods we're comparing to, SCPRED, single cell net, actin, that's on the x-axis. And each of these spots is a different cell type analysis benchmark. And basically, if we are doing better than the other methods, then the green spot should be above and to the left of the line. And if the other method's doing better, it'll be down to the bottom right. And essentially, we kind of outperform these other methods on these tasks. We've got a little precision recall plot where you can kind of see the outer trace is our performance on the right. So we are a bit closer to the top right relative to everyone else. And so our final method is able to distinguish 18 different cell types at the moment. It might be 24 actually. Oh no, it's 24 data sets, it's 18 cell types. So that's available on the website if you need to kind of classify one of these different types of cell. So just moving up to the last bit. Okay, hopefully I won't be too long. This is our kind of current work, things that we're hoping to get finished and get up on the website soon. So my PhD student, Buchan, has been working on a novel protein function predictor. We've got one on the website now, but it's getting on for 20 years old. it's not particularly that accurate, and it only works for eukaryotic proteins. And so what we're interested here is in predicting Go term assignments. So that Go is a kind of common labeling scheme for protein functions. So that's what we're trying to predict. And what we're using here is some pre-trained embedding methods. So embedding methods that other people have worked in. So we use PropT5 as a protein language model. We have PubMedBERT, which is a function text description language model. So we put the textual descriptions of proteins from UniProt into PubMedBERT. We get some features out. We have an embedding called Space for analyzing PPI data, and we use ESM1F for analyzing the structure of the proteins. And we've benchmarked this using the CAFA3 training test set. We can talk about that if anyone's got some questions. But this very hairy, complicated diagram that you don't have to take all of it in, but just looking over here on the left, essentially we have 4 input tracks. So we put a sequence in on the left. In the protein sequence model, we run PropT5 to get a bunch of features out of the other side. And it's the same logic for our functional text. We put the sequence in, we look up that sequence in UniProt, we put its functional text into PubMedBERT, we get a vector of features. And we do the same for structure, and we do the same for PPI data. We go through a common encoder, so they're all compressed to the same dimensionality again, because each of these outputs something of a different dimensionality. And then it all runs through the neural network and outputs Go predictions. The really important thing here is our yellow box, the bilinear gated early fusion. So this is the kind of, I guess, new thing that we've brought here. And what it essentially does, it's a neural network that can learn to weight dynamically the different types of input. So for different sequences, you can attend to different input features more strongly or weakly in order to maximize the Go term prediction performance. And so we benchmark this and we calculate weighted Fmax. And so if we use only the sequence features, that's the top row here, we have these weighted Fmax values. So 0.59 for the biological process Go terms, 0.658 for the molecular function, and 0.692 for cellular component. And then if we add all of the different data modalities, so that's the PubMed text, the structure, and the PPI data, our full model, we see a reasonably good bump in performance. And so for biological process, this, our Fmax, weighted Fmax score rises to 0.611, molecular function to 0.704, and cellular component to 0.706. And the biological process cellular component values there, these are the kind of roughly state of the art relative to the other methods that are using this CAFA benchmark. So our hybrid gated early fusion, so our bilinear gated early fusion is, I think, a really nice way to handle this data where, for different input sequences, different data modalities might be more or less important. One thing that we've got running at the moment, because we've got all of this TED data and all of these protein structural domains, it raises a question about what do specific domains actually do? So we know that in a multi-domain protein, the function of that protein emerges as a consequence of the contributions of each domain. So some domains bind a ligand, some domains might have a catalytic site, some domains are, say, a membrane spanning helix or something. And by combining these together, you get the function of the protein. So like a receptor tyrosine kinase is a consequence of having all 3 of those different domain types. And so if we really knew what specific domains were doing in specific contexts, that raises the question of, well, could we design new proteins by recombining domains when we know the function, like we actually know exactly what a domain is doing in a specific context? And so we've got a little bit of progress here. I've got a master's student who's just finished building a little toy version of our ontology for signaling proteins, for CA and P signaling proteins. But the kind of future project is really to expand out this ontology, this way of describing the domains, such that we can think of them like Lego blocks, and we can build a generative system that will build proteins with functions that we design. So I've got a PhD student going to start on that in September, October this year. And then the last thing that we are hoping to get up on the website, my PhD student McLean has been working on kind of plasmid design methodologies, and particularly like kind of natural language interface for plasmid design. So the the way this works, here on the left, he's kind of gathered all of the plasmid sequences he could find with all of their annotations and metadata. We extract that, and it compresses down to like 100 special like tokens, little descriptors of what features are present on their plasmids. We can convert these kind of descriptors into a kind of tokenized DNA string representation that he uses in his neural network. And then he has a very simple kind of GPT-2 era style model where it takes this tokenized DNA string, this tokenized input, and outputs new plasmid designs. And then the way that works, if you want to make a plasmid, is you describe what plasmid you would like. It generates the tokens. The model generates a group of candidate plasmids and kind of ranks and scores these. And having taken the top ones, it uses the information from the top ones to change the context, the inputs, the prompt for the model. And then we just iterate round and round until the plasmid passes some like quality threshold. And then we can show you a, well, hopefully, quote unquote, well-designed plasmid. And so we have this prototype website that McLean has built just recently. And so here up on the left, there's an option for describing your own plasmid. So you can write, I would like a plasmid with the following features for this expression system. And then it will convert these into a set of tokens, what you just wrote. So here's the token prompt on the left here. If you would like to edit the tokens, you can edit those. Then you press generate plasmid. It does this iterative process and eventually spits out the plasmid. So you get the diagram here for the plasmid that it thinks is best. And you can click on the DNA sequence and you can see the DNA sequence for that particular plasmid. So we're hoping to bring this sort of version of this to the website at some point in the near future. So just to wrap up, I need to thank everyone who's essentially worked on the PSIPRED Workbench over the years. So over on the left here, I have all the people on the left who've contributed code to the web server or a method, particularly Sean, Joe, and Andy for their Marizo and DMP work. Our current group members are myself, leading this particular project, Professor David Jones, who does a lot of method development, Dr. Senwan and Ibrahim Al-Sagaf, who worked on the GSRCL method, Tajan, who works on our TED methods at the moment, and some of our embedding methods, Zishan, my PhD student, who's working on protein function prediction, and McLean, who is working on the plasmids. So yeah, thank you very much. I'll happily take questions if there are any questions.
Thank you so much, Daniel. Very, very interesting presentation. For the audience, you can both ask questions by voice, by unmuting yourself, or if you prefer, you can also write them in the chat and we'll read them out loud for you. In the meantime, let me tell you, Daniel, I think it was like the perfect presentation to choose for the beginning of the festival. Like it's a very, very good introduction to the scenery, let's say, of protein structure prediction and also machine learning tools for proteins. So yeah, it was super interesting. And I'm really excited about the last tool that you presented about plasmid design. I know it's an art on its own to design a plasmid that works well. So I'm really excited to see how this tool may work.
Yeah, I mean, I'm not convinced natural language is the best way. I think, if you're designing a plasmid, you want to know for sure it has the features you want. So maybe just some drop-down boxes where you pick the things you want and it builds it. But the natural language, it's a bit of fun, I think.
Yeah, I mean, at the very least, it could be a good assistant, right? But if you're optimistic, even more. Okay, maybe let me start with the questions while the audience maybe thinks how to ask them or write them down. The first question that came to mind while seeing these different models and the fact that now there is AlphaFold is how do the models in PSIPRED perform in gray areas, like, for example, proteins that have conformational changes? Are they still able to capture I don't know, maybe both the possible secondary structures or for areas that are disordered in proteins?
Yeah, so this is an interesting question. So I think a lot of methods, and AlphaFold is included in this, they're extremely bad at conformational change because, as with all machine learning, what you get is the maximum likelihood prediction. And so if your database, say, the PDB has 100 of one confirmation and one of the other confirmation, then AlphaFold will never predict that other confirmation for you. It's just completely weighted towards the other thing. And this is a truism essentially for all machine learning. So all machine learning methods, and including ours, they're biased to what's most common in the data set. So it's really quite hard to get these things to predict alternate confirmations. And you can see some work on this. But the reality is there's a lack of training data for this. It's because the PDB will be biased towards one confirmation than the other in lots of cases. And even in the cases where you have, a nice 50-50 split, there's, can I think of an example? But like lots of examples where you have things with bound and unbound. And so there's a different confirmation in the bound state, like ligand bound state. There's just not enough of them. Like you need the entirety of the PDB to get the performance that AlphaFold has. And if you think about how many proteins have got two different confirmations in the PDB, that's a small fraction of the percent. So it's a really hard thing to do. And I don't think it will come, I think, I don't think these deep learning methods will do a good job. So I think some more interesting kind of statistical or MD type methods where you take a structure and then use something that's kind of physiochemical or force field based to like perturb the structure and kind of explore alternate confirmations. I think for now, that's the kind of direction that you're probably going to see things if you want alternative, if that's what you want, alternative confirmations. I think if you look at David Baker's protein design work, like he's got bits and pieces where, you're trying to push a model that wants to give you the maximum likelihood prediction. You're trying to push it away from doing that to sample a different bit of the embedding space, essentially. So you're trying to what are some structured ways to perturb that sampling in a meaningful and useful way? But that's very, very hard to do. Yeah, I don't know. I think some of these methods, they're not great for these sorts of things sometimes.
Okay, interesting answer. And another question that I had regarding the hardware needs for your PSIPRED models with respect to AlphaFold. AlphaFold needs GPUs and lots of RAM and takes a lot of time for its predictions. So do you see some kind of research and analysis actually being performed better by the PSIPRED models because of the different kind of hardware that it needs?
So, all of them, usually, oftentimes it's the training of the model that requires the GPU. And so most of our methods on the website at inference time just run on the CPU, and they're mostly small enough to do this in a acceptable runtime. I think the difference, like Marizo search, if you're running it on a fairly modest GPU, it doesn't require a lot. Like a consumer grade, a decent quality consumer grade card. Marizo search will run in about 30 seconds, 40 seconds. Running it on the CPU, on the machines, on the hardware, back-end hardware we run, it's about, what is it? It's about 2.5 minutes. So it's not so great an increase that you have to have a GPU for Marizo search if you download it, but it helps. And most of our models are quite small. They run nicely on a CPU. They're not kind of AlphaFold or I think some of the larger protein language models that like billions or even 1 trillion weights in them. And so you need like 2 GPUs bound together to get these things to run. So we don't have anything like that. I think in the future, I think there's a lot of interest in model distillation to make the models smaller. So once you've trained it, you've got the big model, you use model distillation to make a smaller, faster running model. And so there's what is it called? There's an Apple ML research protein structure model, which I now forget the name of, obviously, where they've taken AlphaFold and distilled its performance. And so it's a really tiny model, but it has the performance of AlphaFold, and it runs, trivially fast. It's a really nice thing, but it is just AlphaFold, but faster. It won't perform any better. It doesn't do anything different. But, I think these sorts of things like model distillation or trying to learn more compact models. I hope / think deep learning is going to go towards more compact models because, you can't just keep adding graphics cards. It's like too expensive, it's too silly, and it costs too much energy, and it costs too much time, and so on. So yeah, hopefully we'll see the rise of more compact models in the future. Yeah.
Thank you. We have a question from the audience by GI. GI, you can unmute yourself and ask the question.
Yes. Hi. Thank you very much for the presentation, Daniel. It was really nice. And I think it is exactly what we need, like a bit of intro and going through each model and look at what it can do and what are the limitations. Yeah. So I think for computer scientists or bioinformatics, usually you guys look at accuracy in terms of like overall the database or basically your testing set, what are the percentage of correctness. Yeah. So as a wet lab biologist, we're more focused on like whether the model is right in our particular case, like the one that we worked for 4 years. So my question is, like, since you work on so many different models, mostly ML based, is there a trend where you can see like what type of what characteristic a model a protein would have that usually makes the model run, like especially the case where the model is run where it is highly confident in its prediction?
Sorry, I'm not sure I quite understand what you're asking after.
Yeah, sorry. So basically, like, is there a characteristic of a protein, like that's whether like it is source or like it is prokaryotic, or that you sometimes you see a trend where the model get it wrong, or like in other ways, like in the ones that the models are run, is there like a characteristic that you can see in that set?
Yeah, you can see these things in some models. So and oftentimes it comes down to like what was the training data, because I think that's really important in these kind of deep learning models is the training. What actually is the training data, and what is the the loss function? What does the loss function actually teach the model? So in the case of our current protein function predictor, PSIPRED, which I didn't talk about, it was only trained on human, mouse, and fly eukaryotic proteins. So it absolutely does its best predictions in those classes. It's reasonably good at other eukaryotic proteins, but it's a little lossy, and it's really terrible at bacterial proteins. Like, it's just not good. And so like we know that method, but that's because like there's intrinsic biases in what we trained it to do. And that's generally what we find for our methods. Like, biases arise because the training data was biased, essentially. We don't kind of see that so much with something like PSIPRED. So PSIPRED is trained just with a very large number of protein structures, essentially, sequences from protein structures, like across all the clades. And it's generally 94% accurate, regardless of what protein you give it. I mean, that just happens to be the case. Yeah, I can't think off the top of my head. Yeah, I think you'll find like the domain segmentation methods. So we have two, DOMPRED and Morizo. Like, DOMPRED is definitely quite biased towards eukaryotic proteins because there are more eukaryotic multi-domain proteins. So like we know that as well. And Morizo search, I don't think has any intrinsic bias in that way, at least not that I know that we've looked at or that we've noticed. But yes, you do find these things, but it'd be very hard. It's very hard to say a priori which method. I think one of our steering committee members, they're also wet lab biologists, and their suggestion for one of our future developments is being able to compare side-by-side predictions where they're different. So that could be quite easy for us to download the AlphaFold DB models and show you that this is where we do or don't agree with some other guys. And I think that would be a very nice way to help people kind of verify predictions, or just compare like where there's some confusion, and then you'd be able to map it back onto what you're doing in the lab, hopefully. So that's a kind of future thing that we're hoping to add into the way we present all of our results, like when there's kind of disagreement between multiple methods.
All right, thank you, Daniel. I guess the question in itself is a bit weird, because I guess if there is a trend that like in your training data that the models always get run, itself would sort of know that and try to correct for it, right? Because so I guess the sort of the wrong predictions are usually like there's no obvious trait for it?
I think in the papers that we produce, we will usually try and make it explicit, like this tool is for this because it is biased in this way. So like FFPRED, we try not to make any guarantees about its it's like archaeal or bacterial performance because we know it was only trained on eukaryotic things. So we try and make that obvious. Like you can use it for bacteria, it's kind of fine, but it's fine in a really generic way. I don't know if you know much about Go labeling, but like the generic terms like protein binding, it's just like every protein binds another protein. And so we can predict that very accurately for bacterial proteins, but like what use is that? Like, you know, that's not useful to anyone. So
Okay. Thank you, Daniel. That's all my questions.
Cool.
Speaking of training data, I noticed that the TED model was trained on the database of AlphaFold.
Which one, sorry?
TED is trained on AlphaFold DB. Am I correct?
For the Marizo search?
Yes.
Marizo method. The Marizo method is trained on the PDB as divided up, as segmented by the cath structural domain database. So the training data is manually verified domain boundaries from the cath database, but it's trained over the PDB. It's not trained over AlphaFold. So we train it over the PDB and then we apply it to AlphaFold. So that's the TED database is the application.
Okay, I see. Thank you for the clarification.
Cool.
Is there any other question from the audience? If not, we are very close to the end of the presentation, so perfect timing. Thanks again for the super interesting presentation. It was a pleasure to have you here. I hope to see many of you in the remaining part of the festival, which has just started today. So the next session is on Friday with 2 more presentations. And let's thank Daniel once more and see you around.
Cool. Yeah, the invite.